Today the topic that Dr. Kniel will gonna be sharing with us, it's Beyond Bacteria, Addressing Viral and Parasitic Hazards in Fresh Produce. This is our call 75 of the Produce Safety Educators Call. So we're very happy that you could join us and you could be here with us. Okay. So instructions, as always, all participants are muted. There will be time for questions and discussion at the end of the presentation, but feel free to share your questions in the chat box. We're going to try to answer to them or hold to them until we finish the presentation. This session is recorded, and the presentation and the recording is going to be shared in our website. And of course, we'll also send it to you, to anyone we have in our listserv. So this list of contacts that we keep, we're going to be sharing this one with you all. Welcome back. I know all of you know that we have had a little bit of a break between educators' calls, but we're happy to have them back. We're going to be hosting a series of these webinars, both in English and Spanish. And the funding provided for these webinars, these sessions, it is coming from the local food safety collaborative. So we're very thankful to them that we can continue with these calls. We know they're very valuable for you. We always appreciate your engagement, your collaboration, you sharing ideas, topics that you want to see. And of course, the participation of content experts that can share this very useful, important information that help us continue our work as educators in produce safety. So welcome back to all of you. For the agenda, of course, introductions. I'm going to be introducing Dr. Kniel, and then we're going to have the Q&A. And please stay after the presentation so you can see some of the PSA updates that we have and upcoming events. So Dr. Kniel, she is a professor for the Department of Animal and Food Science in the University of Delaware. She has decades of experience working in produce safety and food safety. Some of her research is listed here: survival, transmission of pathogens in different complex food systems. She has worked also assessing risks in raw agricultural commodities through water and soil amendments pre-harvest. She has worked studying the correlation between the microorganisms and environmental factors, and also the behavior related to using viral surrogates for this. She has worked also talking of the mechanisms for transmissions of different viruses and parasites through some produce commodities. And she also focused on research and providing these food safety education topics through the One Health approach. Dr. Kniel, as many of you might know, she has been involved in many different associations, including the IFP Association, International Association for Food Protection, where she had served as president and past president. And of course, she has a long time of presenting these webinars, presenting in seminars, sharing her research through numerous papers, books, and book chapters. So we are very happy to have her here, to have her share some of her knowledge with us, and of course, to be able to answer your questions. So thank you, Dr. Kniel, and please feel free to share your presentation now.
Great. Thank you so much, Mariana. That was such a humbling introduction. I really appreciate it. And does that look okay? Good? Wonderful. Yeah. Thank you. So it's really a pleasure to be here today to talk about some of my very favorite subjects. So I want to thank you all for taking time out of your day. So I asked Gemini to help me with an image. And so I thought this would also give us a good way to start today. And I really want to thank again PSA for the invitation to be here, but really the opportunity to share with you a little bit of background about the viruses and parasites, and maybe even get to a little bit of the why we should consider viruses and parasites when we think about produce safety education. So we do have limited time today, so I have included some slides with some additional information that I hope may be useful to you if you want to go back and look at those after we are done being together today. And so I think on this image, you can kind of get a sense of the different organisms that we'll be talking about. Some of the ones that you know very well, and maybe some that you have only heard of just a little bit, and others that are sort of emerging when we think about what to do with food safety and fresh produce. So let's start off with a little bit of the basics, right? Some disclaimers about how parasites and viruses are different from bacteria. So parasites and viruses do not replicate in food. They use the food as a vehicle for their infective stage, be that the virus particle, an oocyst, a larvae, or maybe an egg. That also means we can't replicate them in the food to get more of them to be able to detect. So that's a bit of a challenge. They generally have a small infectious dose, and they're relatively stable to environmental and storage conditions. For example, Toxoplasma gondii oocysts are actually stored in 1 normal sulfuric acid. Can you imagine? Right, so they're really stable. They're quite resistant also to chemical treatments, and this all kind of this story together makes it really difficult to think about control measures. So we're going to talk a lot about prevention at the same time when we think about control. And when we use them in the laboratory or when we're trying to study them, we often have to use animal models or rely on mammalian cell culture. There's also quite a bit of surrogate use in terms of different organisms that we try to understand pathways that might represent one or the other, and they become very useful. So let's think about size. So the organisms that we're going to talk about today range in size from the very, very tiny nanometers in size viruses to larger than your blood cell and things that you can actually see tangibly, like the tapeworms. Right? So we are running the gamut when it comes to size. But because of those difficulties and challenges that I mentioned early on, we're going to keep thinking a lot about risk-based prevention when it comes to produce safety, right? So disinfection-based methods are often very complicated, data is limited or conflicting, and so we often have to think about that risk base. And again, looking back at that size, I really like this image because it shows you some of these differences, even ones you may have heard of, like Cyclospora and Cryptosporidium. Cyclospora is about 3 times the size as Cryptosporidium, and of course, they're all hundreds of times larger than viruses. And then if you were to look at the liver fluke metacercaria that we might find on watercress, it is even hundreds of times larger than it. So let's start with the viruses. As they say, good things come in small packages. So when we think about foodborne viruses like Norovirus and Hepatitis A virus, these are both non-enveloped viruses. And it's sort of a contradictory statement because often we think if you have an extra layer, you must be more protective. But that's not the case when it comes to viruses. Foodborne viruses are non-enveloped, as in the lower panel here, and they actually have what's almost like a puzzle. And if you've ever put together a puzzle so tightly that you could actually like pick it up and hang it up on the wall, that happens in some cases, that's kind of what virus capsid proteins are like. They are that tightly put together, and it allows them to be stable on surfaces and to be transmitted that way as well. Versus something like influenza virus that is an enveloped virus. It has an outer lipid layer that can be neutralized pretty easily by things like hand sanitizer or cleaners. So when you think about that for influenza, right, and as we did with SARS-CoV-2 during COVID, then we can think the opposite is true for something like norovirus and hepatitis A virus, where we know that hand sanitizers are really almost ineffective in most cases. This is an infographic that we created here at University of Delaware during COVID, and I wanted to share it with you in case you want access to it later, but it was very useful for individuals in particular who were trying to convey this information to others, and it shows you when looking at the cellular level what those envelopes are really good for, how they might be involved in binding to a host cell, and then how they disintegrate. So again, you think about that envelope disintegrating and those capsids being like that tight puzzle piece, those are going to be sticking together. And that's why on a surface, we can see transmission by norovirus and hepatitis A virus, where we typically don't see surface transmission for things like influenza. So let's get into a little bit of the details now between norovirus and hepatitis A virus. So I am sure everyone here is very familiar with the disease symptoms like projectile vomiting and diarrhea that's associated with norovirus. Usually 24 to 48 hours incubation, but it can be a little bit faster than that. And then it typically lasts for a few days as well, but you can still be shedding virus even after you have recovered from any symptoms. That's why it's so important that we continue to talk about hand washing and that exclusion of individuals who were sick from jobs where they are actually handling produce, right? Finding another role for those individuals, maybe in the community or in the environment where they're working, if they need to be there, maybe not handling produce, right? For norovirus, you can see there where we look at immunity, exposure does not generate immunity. So you can become infected with norovirus multiple times, whereas hepatitis A virus, which results in a liver infection or which can lead to jaundice. You can have some gastrointestinal illness at the beginning, and you see this incubation period, which is really long, up to 50 days, meaning that our recall of what we just ate can be very tricky for these types of outbreaks. But we do have a vaccine for hepatitis A. It is a vaccine-preventable foodborne illness, which is wonderful, but we have to get those vaccines, and we continue to have discussions about that. There's also some discussion that is occurring globally about how long vaccines are good for, right? We pretty much have good data to show that if we have vaccinations, there's two, about 6 months apart, you have 99% or 98% coverage after the first one and lifelong immunity afterwards. But if you don't get vaccines and you end up getting infected as a child, does that immunity wane? And that's a question that people are asking when we look at endemicity in different areas of the world, right? So that's something just to think about as science continues to learn more and more and more about it. You can see that both norovirus and hepatitis A virus are really important when we think about global distribution, because there are many, many cases of these around the world. So let's look now a little bit more at norovirus in particular. So I love this image. It was adopted from an image from CDC, and it looks at that picture of that hole inside that virus capsid. So you can see where those puzzle pieces are kind of together, and inside it would just have nucleic acid. That's really all they are, which is why you can have this debate as to whether or not they're really alive. But they are so small, and this low infectious dose of about 18 virus particles means on top of a pin, we could put as much norovirus as to make more than 1,000 people sick, right? So kind of a nice image to help us think about it, and something that you might want to convey also to individuals that you're training and teaching. So in 2023 to 2025, I was lucky enough to participate in 2 expert committees where we talked about viruses, first looking at global attribution and then also looking at prevention. Both of these documents are completed. They are available free of charge online. You can kind of Google GEMRA, that's the Joint Expert on Microbiological Risk Assessment, or you can take a picture of this and look those up. These are really nice documents. They have probably more information than you'd ever want, but they have some really great information too on terms of surface disinfection, and that's a question that typically comes up. There's a really great table in this part 2 document if you're interested in that. I'm also happy to help you look up things if you want something very specific, but there's a lot of information there, and it's not all completely, I should say, without contradictions, right? Just like all aspects of science, there are many ways to look at one study. But one thing that did come out of this that I want to point out is while we are talking about norovirus and hepatitis A virus, in particular for fresh produce, there are others as well. Rotavirus, sapovirus, enterovirus, astrovirus, enteric adenovirus, those could all get onto people's hands and be transmitted through fresh produce, although the risks there are really, really low. And if just because of the rarity in terms of transmission that way. So if we look at norovirus and hepatitis A virus, when we analyze this by this expert committee looking at commodity pairs and viruses, something to point out really is this role of frozen berries. And we're going to keep coming back to that. Also, prepared foods, really important for both. So this paper here that I have mentioned, Common and Potential Emerging Foodborne Viruses, published in 2024, has a couple really nice tables in it. This is one of those, and I'm showing this to you here so that you can get an idea of some of the global outbreaks that have happened over the past 20 years. You can see while there are few deaths associated with norovirus and hepatitis A virus, we should note that hospitalizations are not uncommon. And you can see, again, numbers of illnesses per different outbreaks are varying, and you can see a lot of different produce commodities that are involved in these. So noroviruses are non-enveloped, as we said. They are single-stranded RNA viruses, and they're all belonging to the family called calicivirus. And that means they have kind of like cup-like depressions on the outside. They're divided into different genogroups and different genotypes. And so sometimes when you hear about an outbreak, you might hear something called G24. That is the most commonly detected genotype responsible for norovirus outbreaks in people. Again, about 18 genome copies seem to cause illness. Of course, it can be greater than that, and that can vary through transmission route and through the food that might be contaminated. As you can see here on this graph or this graphic, that transmission occurs through the fecal-oral route via direct person-to-person transmission. In fact, that's probably most common, and then also through contaminated food or contaminated water or contaminated fomites. It is not thought to be zoonotic in nature. Although there have been a couple reports that identified human norovirus in feces of animals, but it was probably cross-contamination in the end that led to those, but scientists are still exploring those. So we're definitely think about hand washing and the role of humans in terms of contaminated foods, potential contaminated surfaces, and preparation areas. You may have already seen this before when we think about the burden of norovirus. It's the leading global cause of acute gastroenteritis that results in over 685 million cases worldwide and about 200,000 deaths associated with that when we think about globally. In particular, those deaths occur with individuals over 65 and children under the age of 5. In the US, we have about 20 million cases each year. And the costs to that are increasing because of healthcare costs and lost productivity. And so they're thought to be, of course, in the billions. So it really does us all good when we try to reduce the number of these cases. So people are critical when it comes to transmission of human norovirus. Billions of virus particles can be shed in feces on an average of 8 to 60 days, even in some cases, maybe sometimes even months. There's some varying data in the literature to show that, but peak shedding usually occurs 2 to 5 days after infection. And again, you can have norovirus that's spread easily through feces or vomit or through contaminated surfaces. Again, we think a lot about people, we think about infected workers, and so how do we encourage hand washing, right? That's the best thing that we can do, or helping to make sure that individuals are not in contact with food during that time where they may be recovering from an illness. I also want to add that I think it's important that we have good communication with our workers. I think that's really critical that someone feels trust and safe where they can come and tell you that they have been sick or are not feeling well. So for hepatitis A virus, people are also critical. This is not a zoonotic illness. This is something that is transmitted by people. And so again, when we think about importance for risk prevention, we're talking about hand washing. Really critical here. And thinking about vaccines, maybe, encouraging this vaccine. And as I tell my students in classes, it's one of those vaccines that has some of the least amount of potential complications or side effects. I even got one when I started at University of Delaware working with this, and I was pregnant. And so it actually is a very mild vaccines as far as they go in terms of potential for side effects. And again, you're like 98% covered after the first one. So what is unique about hepatitis A, right? It gets its name because it is a major cause of hepatitis or liver infection. There are many hepatitis viruses. They are not related to each other. So hepatitis A is the only one that's shed by fecal-oral route, and it's very unique that way compared to B or C or E, right? So we think about how important this one is when we think about potential for foodborne transmission. So there are more than 159 million infections globally, with 39,000 deaths, according to the World Health Organization. And from that, about 14 million cases are thought to be related to foodborne transmission, right? So they are shed, again, at high levels in feces, about 100 to maybe over 1,000 genome copies per gram of feces, so less than norovirus, but still quite a bit, and enough to cause a lot of infection and contamination in terms of product. So the timing of virus shedding is really important when we think about hepatitis A virus. So if you see this purple arrow there going across with the star on the right-hand side, that's pointing to that gray block that indicates over this time of infection, you are shedding hepatitis A virus in feces before you even start to feel sick, right? And that's really important, because you might start to feel sick and then say, okay, I probably shouldn't be working with produce. Maybe I shouldn't be, handling some things. I should stay home. But you may be shedding even before that happens, right? And so that's why it's really important, again, to think about good hand washing, just basics in prevention that we're doing all the time to make sure that we reduce the potential for transmission. And so this is also very unique about hepatitis A, where you have this really long incubation period between 15 to 50 days, long shedding in terms of what's happening by the infected individual. And then you can see there you start to wane in terms of any virus shedding. So if we think about hepatitis A virus in terms of decreasing amounts of infections, you can see that from, almost over a decade ago, we really had fairly low numbers of cases in the US. This started to increase in 2017, and you see 2018 and 2019 there. And that had a lot to do with significant increases in homeless populations, especially in California and Oregon, and then spreading from the West Coast across the United States. So just something to think about, that we do have increasing numbers of hepatitis A virus that may be in individuals in the environment. But one good thing that they did when recognizing the importance of homeless encampments and working with individuals is they brought out hand washing stations. And they saw in those specific communities, they were able to decrease transmission. So hand washing really does work. So detection of viruses in foods is pretty complicated. It's time consuming, it's expensive. Here on the bottom of this slide, you can see that there are two standardized methods for this. One is an international standard, the ISO, and the other one is the FDA BAM. They both require specific pieces of equipment and quite, good training in terms of handling the virus, and you may need specific equipment like an ultracentrifuge. So data on contamination levels in foods for hepatitis A virus are sometimes gained from outbreaks, as shown in these two graphs, but it's also really complicated. As you can see, in most of these outbreaks, you see that non-detected in that detection and food category all the way to the right of each of these two tables. And the virus was not detected, even if there was an attempt to try to detect it. So that brings us to this question of, should we be testing foods, right? And I think that that is also not where we want to go, and not where you really want to put a lot of your money, because there just isn't a lot of good methods for that can be simply done. We are continuing to learn more and more about that. And of course, it's done in terms of recalls and outbreaks. But again, that preventative issue is probably the better place to put more of your time and your money. I also want to mention here that in some of this testing, we can't tell whether or not the virus is infectious. So those three images that I have there on the right-hand side, the tests that are being done, which is looking at the RNA, could not differentiate between free RNA, a non-infectious virus particle that is partially disrupted in terms of that capsid, or an infectious virus. And so there are many teams of scientists that are working to try to identify complementary tests that can be done and used in these detection systems to try to help root that out. And I think that that's going to be really helpful for the industry. So water and workers are really where we're talking about when we think about foodborne viruses. And in large outbreaks, the origin may be contaminated water. You may be familiar with these cases. In 2016, there was a large outbreak of frozen strawberries, and those were imported from Egypt. And then in 2017, a large outbreak of norovirus with frozen raspberries that were imported from China. If we go back even further to 2003, poor Chi-Chi's was involved in transmission of hepatitis A virus that was associated with salsa and queso that had chopped green onions. The green onions there could have been contaminated pre-harvest. That's still a remaining question because, of course, it's really difficult to do these investigations when you're finding these contaminated products. And so a lot of studies have gone back to look at it. Could have been that they were iced during production and during sales and distribution. We don't know if that had something to do with it. Or could it be that they were then stored, they were cut and stored in refrigerators and kind of made this like hepatitis A soup with chopped up scallions or green onions? Both of those could have led to, increased transmission, but we don't know, right? And the FDA went in and asked whether or not this could have been something that happened pre-harvest. So lots of questions with where is the origin of these viruses. And those 3 case studies provide a little bit of a peek into some of them. So we keep talking about frozen berries, right? So freezing preserves virus infectivity. This is some work that was done by Alyssa Kelly and Dr. Brianna Anderson-Coughlin in my lab that just shows that hepatitis A virus there on the left and Tulane virus, which is a norovirus surrogate. It's a type of norovirus or a type of calicivirus very closely related to norovirus. And you can see across almost 2 years of frozen storage, the viruses retained their infectivity. So this is actually looking at infection in mammalian cell culture. So what about physical and chemical disinfection methods? There are many of them that have been studied. As I mentioned, there are about 6 pages of a report that has a lot of data. I have a copy here. Happy to send it to you if you'd like it, or you can access it free for that GEMRA report part 2 that came out in 2026. But there's some confusion, right, in terms of if I just want to say, well, how much chlorine do I need to use on a surface? And there's information there for Formica and stainless steel. It's got, this is a really complete table. So I recommend if you have those questions, you should look at that. Brenna Diraxili in my lab is now looking at when you were to add either PAA or chlorine to wash water, what can reduce chances of cross-contamination? Because I think that's a question that's coming up. And looking at what was presented at conferences last year, I think there's a lot of data like this in the pipeline. So I think we'll start seeing more and more papers that will provide information on how much chlorine and how much PAA to use. It does seem that chlorine is somewhat more effective when we look at wash water. And I know a lot of people in the industry are also using that rather than PAA at these high levels. Okay, so let's transition now into parasites, right? These are maybe the forgotten pathogen when we think about fresh produce. And I can say that because from where I'm sitting, I'm really blessed to have good sanitation, right? And most of the growers that I work with on the Mid-Atlantic have really good sanitation. It's not that same way around the world. And because we're a global community, we really want to think about the importance of parasites. I'm also going to provide some information where we look at how changing climates and how different progression of vectors might change the way we look at certain protozoa and helminths. And so it's definitely something we want to continue to think about. As you can see from this slide, we are looking at a range or cast of characters, so to speak, that look really different from one another. We have oocysts that are transmitted here in the case of Toxoplasma, Cryptosporidium, Cyclospora. These are kind of egg-like, but they're not quite eggs. And those contain infectious life stages. I'm not going to talk about Giardia here because we don't, at least in a lot of the surveillance work that we've done and I've seen more recently, it's important, but it's not something that we see too frequently, but you might recognize if you've seen before these trophozoites, they have a really unique look to them. Trypanosoma cruzi, which causes Chagas disease, is something that we are becoming more concerned about with fruit and sugarcane. Fasciola is a liver fluke, and Echinococcus here, this is an egg of a tapeworm. That's becoming increasingly important when we think about fresh produce. So again, we're going to talk about prevention because that's really where we can put solid footing, right? So protozoa, roundworms, tapeworms, and flukes, all important foodborne pathogens. When we think about how best to inactivate them, heat, thermal inactivation is going to be key. Detection is often including some type of molecular detection looking for the nucleic acid. And so we have the same problem that we had with the viruses. In some cases, we can't tell if they're viable or not. That's going to be really important when we talk about Echinococcus, kind of at the end of this talk. Also, concentration from foods is really difficult. If I'm detecting one egg or one oocyst or 10 oocysts, right, that might be my limit of detection in terms of what I can detect, but that may be all that's there. And so I can't easily concentrate them or grow them up like I can for bacteria to be able to better detect them. Also, they're not always apparent, like this lovely couple here that apparently has worms. We don't necessarily see that all the time. And we may have asymptomatic individuals. Or a lot of times we have such a low threshold, right? We don't know it until there is an outbreak or it gets missed. So when we're going to talk about One Health as well, because we want to think about the role of veterinarians in this and good communication kind of across our healthcare spectrums. So in this flow chart here, you can see if we look at our crops there in the center, we have lots of potential inputs where contamination can originate. We have surface water, we have runoff, we have sewage sludge, where at least in this area of the US, I hear about potential point contamination issues quite often where, this aging infrastructure that we have for sanitation or potential runoff or something that occurs with wastewater plants, right? So we have to be aware of those things, especially when you think about that agricultural risk or water assessment that we are talking to our growers about. So let's start by talking about Cryptosporidium parvum. So at the bottom of this slide, you can see this life cycle. And as food safety educators, when we think about these complex life cycles that parasites have, I want us to recognize what that means to us, not as parasitologists, but as produce safety educators, because that means that this delay in infectivity or a delay in the host response to that infection can also lead to a delay in knowing when someone is sick, and then in the ability to have a recall, in the ability to understand that there's a contaminated product. And so that is adding weeks or months to our timeline, right? And that's really important to us because we can't identify contamination very quickly. So Cryptosporidium parvum and Cryptosporidium hominis are two specific species that can infect humans. Cryptosporidium parvum is actually zoonotic in nature, so we see transmission from cattle or deer. Hominis is only shed through people. And in some cases, actually, there's other Cryptosporidium species that can make people sick, especially when we see individuals who are immunocompromised. You can have infection by Cryptosporidium species that are in reptiles or in birds or turkeys or rats or those types of things. There's basically a Cryptosporidium species for almost every animal living. And they look like these little oocysts here. This is a microscopic image of them. There are these oocysts, these tightly packed oocysts that once you ingest them, they make their way into your intestine and they exist, or these banana-like creatures called sporozoites come out of those oocysts. And then they make themselves this quite comfortable little home, which is actually called a parasitophorous vacuole. They're actually sitting outside of your intestine. So this is an image that I took in graduate school looking at infected human intestinal cells. And you can see this sitting here. And this has all these little life stages inside. And if you notice, these are all the remnants of your microvilli that are usually upright and beautiful and all ready for nutrient absorption, but Cryptosporidium has blunted all of those, and this is why this infection results in a really profuse watery diarrhea that can last for weeks. if you're healthy. If you're immunocompromised, if you notice this little thin-walled oocyst rupturing here, about 20% of oocysts that will be shed, and I should go back a step and say there's asexual reproduction followed by sexual reproduction, so then the human host will shed these oocysts. So about 20% are thin-walled, which means that these guys are actually going to auto-rupture in the human host. And if you can't mount a cellular immunity response that's strong enough to be able to recover from those, that is where we see individuals succumb to this infection. So it's really quite serious. So if we look at food net data from over time, we can see that cryptosporidiosis really has been increasing, right? And so you can see this kind of dashed brownish line that's going up there. We don't have any more good data that shows this. I'm sure that it had some, like a roller coaster, and it's probably down quite a bit, but interesting that it went up so much there. Why did it increase? Well, since it's a zoonotic agent or organism, it definitely can be coming from different places. We also want to note that Cryptosporidium is incredibly resistant to normal levels of chlorine, meaning that it is really it's an organism that can definitely be shed and be shared among individuals in recreational water outbreaks. You can see in 2016 there, we had quite a bit of recreational water outbreaks. You may have also heard of this organism because it was tied to outbreaks with apple cider and really important in terms of juice HACCP. And so it's something that's mentioned there, but it is resistant to normal levels of chlorine. So it's a really interesting organism that way, and it has to do with that tough oocyst wall. So another organism with a tough oocyst wall that you probably are quite familiar with is Cyclospora cayetanensis. This is actually a non-zoonotic, so this only infects humans. So when we think about outbreaks of cyclosporiasis, we're looking to where contamination originated from humans. We have been learning about this organism for the past couple of decades for sure. It's originally a waterborne parasite. The oocysts are different from Cryptosporidium. So you can see here, it goes through asexual reproduction similar to Cryptosporidium with these sporozoites that infect the intestinal cell. But then we have what's shed is called an unsporulated oocyst. So basically it's immature and it needs to be in the environment. It has to have the right conditions in terms of moisture and air, and then it will start to sporulate. And you can see that changing here. So it's this mass that's undifferentiated inside, and then it starts to become differentiated with two sporocysts that contain the sporozoites. The exact mechanism that causes this is not really well understood. Part of that is because it only infects humans. We have limited organisms to be able to work with. And so there are scientists in the US that are working on this. It's a fairly large organism, so bigger than crypto, about 8 to 10 microns, where crypto is just about 4 to 5 microns. Also kind of interesting, especially in the original studies that are done using microscopy, this autofluoresces. So you can see that you can actually detect them there by microscopy. And of course, that's what's was done for decades. Now we're looking at the detection that has really evolved, and we have some really sophisticated detection systems that have been published by FDA. So really nice work there in terms of the ability to detect in different produce commodities and also in water. So symptoms of cyclosporiasis are that watery diarrhea, your basic, everything that goes along with that. It can last for several days or even weeks. It affects both immunocompetent and immunocompromised individuals. The outbreaks have shown that we really don't have deaths associated with this organism, but there are increasing numbers of hospitalizations. And so that's something to consider. It can be treated with the sulfa drugs, but if you are allergic to those, then there isn't really a good treatment. So some key information that we've gained from cyclosporiasis outbreaks, because as you all know, this is a great place to learn more about these organisms. So starting in the 1990s to the 2000s, we had many outbreaks that were associated with imported produce, berries, snow peas, herbs, lettuce, and some really interesting outbreaks. If you like case studies, some of these are fabulous because you're looking at the basil in the vinaigrette, right? And who knew that there was basil in the vinaigrette? Or the raspberry coulis on the cake plate, right? That's not a good thing to happen at your wedding or your baby shower. And that definitely has occurred. So unfortunately, it's a shame, but they make great case studies. So in 2013, we started seeing these annual outbreaks that happened associated with leafy greens and herbs. And in 2019, the CDC started keeping track of a separate line of cases that are referred to as domestically acquired. So these are individuals who have confirmed cyclosporiasis that do not have any international travel and are not part of an outbreak that might have happened that same year. I should also show you here, I have included this one paper that talks about the investigation and the environmental investigation that the FDA did, looking at the different plants, the processing plants that were involved, and going back and looking at the produce that was grown involved in this 2013 outbreak. And you can see this long epidemiological curve here on the bottom that's part of this outbreak. So if you're looking for some fun reading, it's a great paper. So if you're curious about where we sit today, so in 2025, there was just over 1,000 cases that were confirmed. So again, we're seeing this every year. It's not going away. So what do we do about it, right? That's the question. So currently our testing methods, as I mentioned, are primarily restricted to research and for clinical typing in epidemiology. The CDC has an outstanding group that's doing a lot of really interesting work in typing, looking at relationships of these organisms, and the FDA has done a really great job in terms of what they're doing, evolving the type of testing that is being done, and making sure that it can be used for various produce commodities. It remains based on DNA detection, and in my perspective, it's not something that we need to be doing on the farm because we just are not quite there yet. So really saved for recalls and outbreaks. So what do we do then, right? Well, there's been some work and increasing work on filtration because these oocysts are so large. They can be removed in some part by sand filtration. And then the USDA ARS group led by Manan Sharma and some work with Michelle Danilek at University of Florida have looked to see whether or not that can be scaled up and be used in produce fields. So we can see some removal of our surrogate organism for Cyclospora, which is Eimeria, which is about the same size and same shape, similar in terms of what is on the outside of that oocyst as well. So a good surrogate there. If we're looking at what can be done if we need to maybe divert produce that's contaminated, one option that is available, of course, there's thermal processing. It's probably the best, but we also have done quite a bit of work with high pressure processing of food products. So all of those cold pressed juices that contain apples or any kind of apple product also have to show that Cryptosporidium could be inactivated during that process. And so that's definitely something that can also inactivate the oocysts. So again, when we think about what can industry do for Cyclospora, because it is a concern for the produce industry, right? Attention to hand washing, sanitation, what's happening with the restrooms, are people using the restrooms appropriately, is toilet paper being put into the right receptacles, not allowed to blow around, because these oocysts can survive really long periods of time in the environment so they could get out into the soil. So really kind of trying to understand more about that in terms of its environmental persistence and really understanding those triggers for sporulation that are not well understood. So the next organism to talk about is Toxoplasma gondii, which I'm sure many of you have heard of, and you're all thinking now, oh, that's the one with the cats, right? Yes, so Toxoplasma gondii is the second leading cause of death due to foodborne illness. It is high mortality, but we don't see so many cases, so I don't want that to make you too nervous. It's also a bit of a misnomer. So the cat is the definitive host, meaning it is the only animal that's going to shed the oocysts. But cats are not always shedding oocysts. So this diagram here comes from the Cornell Feline Health Center. They have some really nice information if you're concerned about that. One of the best things that you can do is not let your cat eat live animals like rats or birds, because that's where they could become infected. Also, if you have a healthy cat, it's unlikely to become infected. If it becomes ill with feline leukemia or something like that, just like us, it has it's immunocompromised, it can then become more infected or start shedding, especially if it's eating outside like birds or rats where it comes in contact with the oocyst. But these oocysts are shed just by the definitive host. So this organism is interesting because it can cause a flu-like symptom, but it's not gastrointestinal. And when we think about what's happening to humans when they get infected, we actually can get tissue cysts, where the organisms that propagate from the ingestion of the oocyst or ingestion of an undercooked tissue cyst can migrate into the muscles of the human. They also migrate here into these animals that we raise for food production, right? So 3 ways to get an infection from Toxoplasma gondii: to ingest the oocysts in fruits or vegetables. They can also be transmitted in water. I have air there mentioned because there have been conditions that were really dry around a farm that had a lot of feral cats, and they saw potential for transmission that way. Or if you ingest a tissue cyst in undercooked meat. And this is not so much a concern for our animals that are raised in conventional agriculture, but more the wildlife and the hunting, those animals that are a little more exotic, like boar or cougar. So you want to make sure they're cooked very well. And I think most tragically is congenital transmission, and that occurs when an infection is acquired during pregnancy. So my little PSA here from my own little soapbox is that if you are a woman and you are thinking about having a baby, or you're have a chance of getting pregnant, just know whether or not you are positive or negative for Toxoplasma. If you are already seropositive, you're kind of protected. You won't seroconvert during the pregnancy. When you're pregnant, ask your physician about it, because they won't always test you. So it's really nice to kind of keep track of that. They do that a little bit better in other countries than we do here in the US. But in terms of cats, for your ag water pre-assessment water assessments, you may want to consider thinking about whether or not you have feral cats, and is there a chance to have runoff. So this is another life cycle that you can see here. Again, the oocysts are going to be shed from the cat. They're shed unspoilated or undifferentiated. They need that time in the environment. And this is where produce can become contaminated. Otherwise, these animals may ingest oocysts in fields and forage, or they can ingest tissue cysts. For example, rats or birds are sometimes caught while mowing hay, things like that. And so they may be ingested accidentally that way. So best advice to always cook your meat well. So I want to also mention that we're seeing some changes in terms of risk in animal agriculture, as more and more individuals are interested in having animals that are raised by these sort of non-conventional methods, and looking at free range and free pasture raised chickens and also swine. So there's a whole host of CDC recommendations. I'm going to skip this and let you look at that so I can make sure we talk about the last couple of organisms here. So Chagas disease is interesting. And up until a few years ago, I would not have really been talking about it when it comes to foodborne transmission. But what they're seeing in South and Central America, and what is being done and work at the University of Florida Emerging Pathogens Institute, I want to make sure that we do mention it. So these kissing bugs, or the triatinamine bugs, there's a picture of one there. They're about the size of a penny or a nickel, so you can see them. They're big bugs. These are the bugs that can carry the parasite called Trypanosoma cruzi that causes Chagas disease. So that is usually transmitted as the bug bites and gets into the infected feces, then gets into the human. But what's interesting is that we're starting to see where the bugs or the feces are getting into fresh produce or on sugarcane. And when individuals are ingesting that, they get a much higher parasite load. And so perhaps that higher parasite load all at one time through food is leading to fairly severe symptoms. And so these symptoms are something like acute myocarditis, or meningoencephalitis. So they can be quite serious, and about 8 to 35% mortality rate compared to like 5% if they're a vector transmission. So definitely something that we want to keep an eye on, especially if you are working or growing produce in the warmer regions in Florida or in South and Central America. That's really important that we continue to think about that. So a couple images here for you on the life cycle. So the most common type of foodborne transmission happens when kissing bugs contaminate food and drinks directly. Usually this happens if they defecate in the juice, or there could be some of these nymphs, the larvae, that are they're smaller, they're about a few millimeters in size. They could actually be incorporated into a food product. Sugarcane juice, ice cream, and shrimp has also been seen to be a vehicle. But there's scientists here at University of Florida that are really looking to see what happens in terms of animal association with this, because there can be contamination and infection if you eat undercooked meat, or in some of blood from these animals that might be used for religious rituals or traditional medicine. So again, a lot of people are trying to ask good questions to understand this transmission route. This comes from this great paper that was looking at what's happening in Colombia and specifically asking the question of where is the highest risk? Is it from direct ingestion of the bugs, as shown on the right-hand side, or cross-contamination with the feces? And that's really important when we think about what information we might provide to growers and harvesters when you're thinking about these bugs being present in sugarcane and in fruits and vegetables. Okay, so the last couple things that I want to talk about have to do with helminths, these larger worms and flukes. Soil transmitted helminths are actually a really big problem globally. Again, we don't see as much of an issue in the US, but a few of them that I wanted to mention. And we're going to talk about this from a One Health perspective, right? So our best control measures that we have, of course, are thermal treatment, peeling or scrubbing, where you can actually rub off the larvae or the eggs that could be on fresh produce. There's a lot of studies that are looking now at chemical soaks, acetic acid soaks, or a 1 part to 2 part vinegar plus vigorous washing. So the stream of water is really important to help remove the larvae, or ultrasonic cleaning, which is probably not so useful to most growers. So Angiostrongylus cantonensis is the rat lungworm, and this is a parasitic nematode that was first described in 1935. We've had about 3,000 cases in humans since 1960. So not in terms of risk, right? Not huge. Most of those came from Thailand, where rare snails are consumed as a delicacy, right? Kind of like if you remember Ronald Weasley eating slugs in Harry Potter. So we're trying to avoid that. But in cases here, you might have this snail, which is the intermediate host in terms of this organism, and can actually shed the infectious larvae in the trails that you see sometimes when they move along produce. And so what we want to do is try to reduce the presence of those snails, or the Hawaiian Department of Agriculture actually has a lot of really great information on that, because this is where we're seeing it predominantly. I was actually lucky enough to visit on spring break recently, and we saw lots of snails. And every time we went, I said to my daughter, maybe that's Angiostrongylus. She was not too impressed, but I thought it was kind of cool to see them all over the place. They're beautiful creatures, but you don't want them to be on your produce. So again, there's some data that shows that a 1 to 2 ratio of vinegar to water, a soak with a vigorous rinse, and then of course doing that visual slime check. So Fasciola is also kind of an interesting organism. This is the liver fluke. We see these have to do with these these hosts are either cattle or swine, right? And then we serve as a host as well after we are consuming one of the larval stages. So not the large fluke, but the large fluke then can develop. The vinegar does not work so well on these, so you don't want to use that on your watercress. Quick case study on this. This was shared with me about 15, maybe 20 years ago, where an individual in the US was consuming quite a bit of watercress because he saw that it had a lot of health benefits. This was locally grown in Florida at a location where they had everything you need to have fasciola. They had watercress, which was grown close to the water in a very moist environment. You had cattle and you had snails. And then in the same place, we actually had poor irrigation and sanitation. And so there was this really great area for the snails to be able to have all of their metasarcaria, which is one of the larval stages that binds to the crop. So again, you can see those, and you can also look at systems like this where you'd want to promote better sanitation. Lastly, I want to mention Echinococcus. So again, not on my radar for a very long time, but something to think about. We are seeing changes in terms of the spread of their host, dogs, coyotes, foxes, that in this map here, you can see in these darker colors are where Echinococcus was detected, right? So this is called, it has an incubation in humans that consume eggs that are in feces of foxes or coyotes. And you will not know that you are sick with this for maybe 10 to 20 years. And so it's something that we are really trying to watch and learn about. And a lot of veterinarians are studying this. And even a couple of years ago, well, 4 years ago now, we saw actually a food safety news article come out and talk about the DNA from Echinococcus being found on berries. So again, DNA, we don't know if that's a viable organism or not. So control and risk reduction for a lot of our helminths and parasites, right? Pre-harvest is the most critical, using microbiological fit for purpose irrigation water, avoiding untreated manure and wastewater, really using that One Health approach when we think about animals that might be in the region or nearby or on the farm. Post-harvest, you can remove some eggs and larvae, may not be fully effective, so working on that pre-harvest instead. Freezing is great for helminths, for the worms. You can absolutely freeze product. Cooking, thermal is always great. And then at the consumer level, right, thinking about washing hands for those viruses too, and just being more and more aware. I always suggest that we have a focus on people, especially when thinking about norovirus and hepatitis A, and relying on those good agricultural practices, and all of you who are doing excellent training in this area. And then just to leave us with some food for thought, right? There are a lot of gaps in terms of detection methods, understanding the viability for some of these organisms. We need specific guidelines globally to promote better food safety around the world. And then as we are watching with global trade and climate change and anti-helminthic resistance, that can complicate some of the control with some of these other organisms. So sorry that I went over a little. It's too much fun of a subject. I apologize.
Thank you, Dr. Kniel. No, it is great. A lot of information about many different viruses and parasites that we need to keep in mind when talking of product safety. There's one question about how does free-ranging animals like chicken and swine impact the viral load?
Yeah, that's a great question. So when raised conventionally, like on cement floors, there tends to be no access to things like coccidia or oocysts or eggs or helminths or tapeworm pieces or things like that. When thinking about toxo in particular, it's out in the environment. And so these animals end up coming in contact and eating those in their grass and while they're foraging. And then depending on the organism and what they ingest, they can become contaminated that way or infected that way.
Great, thank you so much. And then there was a question from the registration. I think you talked about rat lungworm, and that came from that. Thank you for that. But there was a question about sanitizers. If there's better sanitizers for viruses and parasites, and also the discussion of concentrations.
Yes, so lower concentrations of chlorine do seem to be fairly effective. Now, if there's a specific question about a water or a surface, I'm happy to help look that up in terms of the time. Like 1, 5, and even 10 minutes can get you inactivation of viruses on different surfaces and in water. For some of the helminths, chlorine seems to be better than PAA, but then also because they're so large and they do all have this stickiness to them, the oocysts and viruses and the eggs and larvae, the scrubbing is always really a nice thing to add to any kind of, like, if you're trying to reduce the potential risk.
Thank you. Thank you for that. And Dr. Kelly Kniel shared here an email. So if you have any more questions or comments for her, please feel free to email her or send them out to me, and I'll make sure that she gets a summary of any questions or comments you have about viruses and parasites for her to illustrate to us. Again, thank you. Let me share just really quickly a update. As many of you have seen probably in our annual update, we are working hard on V2 pilots. We'll be hosting training shortly. Thank you to all those who submitted the survey information that you were willing to participate in these pilots. Our goal is to complete this V2 by August 2026. And then we currently have no funding to launch these V2. So please wait for that part. But PSA team update also, as you might have heard before, we had to reduce our team. We have this decreased by 3 people. None of us are fully funded by the current cooperative agreement. So we had to come up with some other projects, as you see, to fund us, the ones that we continue working for PSA. But of course, we continue focusing on accuracy and high level, high quality information for all of you. And then about the next call, we're going to have our very own Dr. Bean sharing about crisis management. And it's not just about food safety. So this is going to be our next call. Please be on the look for our registration link. As always, we're going to share it through the listserv and through the different social media channels that we have. If you happen to know about this call on any calls through social media, but you want to get the first invitation in your emails, please join the Pro Safety Educators group. Here is a QR code where you can scan with your phone and fill out the registration information, so you will have all these emails ready for you. And then if you have any input on what you want to hear, what you want to see in this educators call, please just send me an email with your topics or suggested speakers that you would like to see on these webinars. Thanks again to everyone who is joining, and sorry about the 3 minutes that we took of your time. Thank you all.